Hello friends, today we are going to discuss one of the important theme in biomolecules chapter, DNA replication. DNA replication. What is DNA replication? DNA replication is a process in which DNA copies itself to produce identical daughter molecules of DNA. And the fidelity of DNA replication is most important, of course, essential for the existence of life. Because during replications, a mismatch may affect the genes, that means the quality of genes, that means the way of mutation, and leads to a abnormal character. Say if it is a protein, an abnormal protein, if it is an enzyme, an abnormal biochemical character. So that means the existence of life, the very existence of life depends on DNA replication. So the accuracy is quite important here. Okay. And DNA replication in both prokaryotes and eukaryotes is semi-conservative. The kind of DNA replication is semi-conservative. We discussed that is nothing but production of or producing identical copies of daughter molecules of DNA. Okay. What exactly is semi-conservative? In semi-conservative DNA replication, the resulting daughter DNA molecule contains one strand, which is a parental strand, and one new strand. That means since half of the DNA copy has been, that means preserved in the way of parent, a copy of parental DNA has been conserved, so it is called semi-conservative. For understanding, in the form of an illustration, say it is a DNA molecule. For understanding, we say it as strand A and strand B, and it leads to replication. Produce two identical daughter molecules of DNA. Say here A P and B D, A D and B P. Here P stands for parent, D stands for daughter. That means these are the daughter DNA molecules, D1 and D2. This is parental DNA molecule. It produces two daughter DNA molecules, wherein half, that means one strand coming from parent, here A, and here it is B. The other strand is a new strand. That means half, semi-conservative DNA replication. The thing is, the DNA replication in both prokaryotes and eukaryotes is almost similar, except in the kind of enzymes and origin of replications. We'll discuss point by point, okay, for better understanding. For conduciveness, we'll discuss about the importance of enzymes involved in DNA replication at first, followed by the process, okay. You know, any biomolecular activity is highly dependent on enzymatic activity. Same here also. And different enzymes involved in DNA replication are the most important enzyme, DNA helicase, DNA single stranded binding, DNA binding protein, then DNA polymerases. There are three kinds: DNA polymerase 1, 2, 3. Apart from these, there are other enzymes like topoisomerases. They are again of two kinds: topoisomerase 1 and topoisomerase 2, otherwise called gyrases. DNA ligases. These are the different enzymes involved in DNA replication. DNA helicase, in a sequence, we'll start with DNA A. DNA A protein separates the two DNA strands. DNA helicase, it opens up, that means it is a continuation of a replication bubble. So the first activity is DNA A, it forms replication bubble, and DNA helicase or unwindase destabilizes the helical activity, just like a zip, the activity of DNA helicase. Then to maintain the unwindase activity, that means to keep the two strands open, the single stranded binding protein SSBA, otherwise called DNA helix destabilization protein, will hold on into two individual strands so that it shall not wind up again. That means it shall not get into the helicase activity again. That means it should be kept open. Then DNA polymerase, the involvement of DNA replication is by means of these DNA polymerases. DNA polymerase 3, it is the DNA replication enzyme. This is very important. DNA replication enzyme. Whereas DNA polymerase 1, it will remove the RNA primer. DNA polymerase 2, it is a DNA repair enzyme. Then topoisomerase, they avoid supercoils, tension activities. No? DNA ligase, they form phosphodiester bonds between successive DNA fragments. I will tell you how it happens. Okay, it's about different enzymes and their role involving in DNA replication. Then the process. DNA replication is a bit complicated process because of certain activities, because of some setbacks. I will tell you what exactly it is. The first foremost step in DNA replication is initiation of DNA replication. Initiation of DNA replication. That means ORI. Origin of replications. They are nothing but AT base pair rich portions. Number 2, helicase activity. And other enzymes. Number 3, RNA primer. Number 4, semi-discontinuous and bidirectional replication. Number 5, topoisomerase activity and proofreading by DNA polymerases. These are the broad steps involving in DNA replication. The first, initiation of DNA replication, helicase activity, RNA primer activity, semi-discontinuous and bidirectional. This is very important. And 5, it's called error management by means of DNA polymerase in proofreading activity and topoisomerases. First, here, initiation of DNA replication. We have to identify there are certain zones, certain points in DNA where the exact replication starts. They are called as ORI, called origin of replication. I told you there are so much similarity. I can say a close resemblance between prokaryotes and eukaryotes with respect to DNA replication, with certain differences. Say there is only one ORI in prokaryotes, but there are many ORIs in case of eukaryotes. But with just one origin of replication. How to identify that this is ORI? Because there it is 80 base pair rich portions. And this 80 base pair rich portions, the first, I told you already, DNA A protein. First we will discuss, we will summarize the process, then through a diagrammatic illustration we will explain. Okay. Then 80 base pair rich portions are the sites of ORI, that means origin of replication, to which the DNA protein binds and it separates the two DNA strands. Then the helicase activity, that means the helicase enzyme binds to the double stranded DNA and opens up, just like zip. I told you, just like, say these are the two strands of DNA, just like zip, that means it releases the helical activity. helicase activity. And here then other enzymes, here the SSBs, I told already, single stranded DNA binding proteins, otherwise called DNA helix destabilization proteins. They also assist in DNA unwinding activity. That means after the DNA helicase go along the strands, we have to ensure that DNA shall not get into again helical activity. So it had to be hold tight, and this hold tight activity is done by SSBP. They don't involve in catalytic activity, but just hold the strands tightly so that it cannot get into helical nature. Okay. Then RNA primer. After that means it is in the way of replication bubble and replication fork. When this happens, it leads to replication bubble and replication fork. And when the replication fork opens, the RNA primer activities comes. You know, DNA cannot synthesize directly on its own, but there should be a precursor called RNA primer. RNA primer is synthesized by primase, a RNA polymerase plus some other enzymes, say single stranded binding proteins, to form primosome. This together, that means the primosome synthesizes the RNA primer, which is nothing but some 5 to 50 nucleotides length. Then, this is very important. How the DNA replication starts? Whether it is homogeneous? No. There are certain setbacks, I told you. It's quite complicated. Why? Because the DNA replication takes place only in 5' to 3' active or 3' direction. Because there are no enzymes that can synthesize DNA in 3' to 5'. That means there are no DNA polymerases. That means DNA replication is synthesized by DNA polymerases. Specifically DNA polymerase 3. DNA polymerase 3 is involved in DNA replication, but only in 5' to 3'. There are no DNA polymerases that could synthesize in 3' to 5' direction. Because of this setback, DNA replication done in two different ways. That means it is called semi-discontinuous. I told already, there are two strands, if I understand. One strand, this is called leading strand, and another one is called lagging strand. The leading, the name it implied, is having 3' end. That means the corresponding complementary strand should be 5' to 3'. I told you already, DNA replication done in 5' to 3', and DNA polymerase 3 will simply add the deoxyribonucleotides in 5' to 3' direction. That's it, over. But in case of lagging strand, that means having 5' OH and should add that should continue or should stretch the DNA strand in 3' to 5' direction. There are no DNA polymerases. Since there are no DNA polymerases, it should have an alternate path. Here the thing is, in case of leading strand, the 5' to 3' direction of DNA polymerases takes place towards the fork, whereas in case of lagging strand, the DNA polymerase activity again only in 5' to 3' direction is away from the fork. To avoid this uncertainty or to avoid this problem, simple, the DNA replication on lagging strand will take place in 5' to 3' direction away from the fork. Whereas in case of leading strand, 5' to 3' direction towards the fork. But the thing is, in case of lagging strand, DNA replicates in a discontinuous manner in a small fragments called Okazaki fragments. It is named after the Japanese scientist Okazaki who reported this process. And the credit ultimately goes to Messels and Stahl, who have first given the experimental evidence of semi-conservative DNA replication in 1958. Since the lagging strand synthesized in a discontinuous manner, it is called as semi-discontinuous. Since the directional, that means the replication takes place in both the directions. From the replication bubble, it is called as bidirectional replication. So, semi-conservative, semi-discontinuous and bidirectional replication. These are the important three words associated with DNA replication. Okay. After the replication is over, that means during the activity, there are topoisomerases. Because of this unwinding activity of DNA, the tension may generate. It may lead to formation of supercoils, and the supercoil formation is avoided or prevented by means of topoisomerases. And after the DNA replication, along the DNA replication process, the mismatch base pairs shall be avoided. That's what we call proofreading by DNA polymerase 3 itself. And at the end, the RNA primer, which actually contains ribonucleotides, shall be removed by DNA polymerase 1, and the two DNA strand, that means the small DNA fragment and the large, that means the continuous DNA fragment synthesized by DNA polymerase 3, shall be attached by means of DNA ligase. Is the overall activity. So we will discuss the same by means of a simple a self-explanatory diagram for better understanding. Say this is a native DNA. This is native DNA, and this is called DNA A protein. I told already, DNA A protein will open up the two strands. Then it forms replication bubbles. Replication bubbles. Then next step, a replication fork. This is very important. As I told you, this is the entire process has to be illustrated here. Say how it works. I told you DNA helicase like act as a zip opener, like a zip. This strand is 3' to 5', and this strand is 5' to 3'. The 3' to 5' strand is called leading strand, whereas 5' to 3' strand is called lagging strand. And this is DNA helicase, and these are, I told you already, there are single stranded DNA binding proteins, SSB, simply called single stranded DNA binding proteins. Then the leading strand will synthesize in 5' to 3' direction. Say this is RNA primer, and this is DNA polymerase. This is RNA primer synthesized by primase. Then it is towards it is replication fork. Okay. Then the lagging strand, since there are no DNA polymerases that could synthesize in 3' to 5', I told you already, it is away from the fork. Say the RNA primer should be the initiator, and this is DNA polymerase. Then another RNA primer, then DNA polymerase. In the way of small fragments, small fragments. These are RNA primers, and they are called Okazaki fragments. And the direction is away. Okay. It's a simple representation. First, with the native DNA protein will bind what we call ORI regions. And these ORI regions lead to opening of strand, forming replication bubble, and the replication bubble further opened by replication fork by means of helicases activity, DNA helicase or unwindase. These DNA helicase, they go along the strands, two strands, that leads to a zip, what we call zip opener. And after the zip opens, that means SSB, single stranded DNA binding proteins, otherwise called DNA helix destabilization proteins, it will hold the strands tightly so that it will not get into helical activity or nature. Okay, and after this ultimate action, that means the DNA polymerase will come. DNA polymerase 3 we have already discussed. DNA polymerase 3, first here the two strands simultaneously they duplicate. This is very important. Simultaneously duplicate and is bidirectional, because from the replication bubble it will go in two directions. That means one is away, one is towards. But at 5' to 3' one is away and towards. That is what is called bidirectional. Okay, and how it ends? First the RNA primer will come. This is RNA primer. Synthesized by primase, which is nothing but a RNA polymerase, a specific RNA polymerase. After the RNA polymerase synthesize, the DNA polymerase 3 comes into action. In case of leading strand, it will synthesize in 5' to 3' direction towards, towards the replication fork. Here the thing is, mostly one RNA primer is enough. One RNA primer is enough. But in case of lagging strand, the issue is different, because there are no DNA polymerases that could synthesize in 3' to 5' direction. So it has to depend on alternate mechanism, 5' to 3', but away from the replication fork in small fragments called Okazaki fragments. Then later onwards, DNA polymerase 1, they are called gaps. DNA polymerase 1 and ligases, they attach different Okazaki fragments to form a continuous strand of DNA. Then after that, RNA primer, which actually contains ribonucleotide, it has to be replaced. What we call NIPS. Nipping means removal of RNA primer, simply called NIPS, or a replacement of RNA primers by corresponding complementary small fragment of DNA. Who will perform this? The DNA polymerase 1. So DNA polymerase 1, this replace RNA primer, and this RNA primer, just imagine, this is the long stretch of DNA synthesized by DNA polymerase 3, and this is RNA primer. It will be replaced by appropriate DNA fragment by DNA polymerase 1. Then they add them together by means of DNA ligase 1. That means DNA ligase, which is nothing but an enzyme that form phosphodiester bonds. Esterification shall be done by this DNA ligase, and helicase and DNA ligase activity are energy consuming enzymes. For example, DNA ligase ATP leads to AMP plus PPI. The same time helicase is that means energy demanding enzyme. Then since RNA primer is removed, therefore a continuous strand of DNA is done. And after that, during this process actually, topoisomerases, I told you already, topoisomerases avoid supercoils, tensions, particularly topoisomerase 2, always called gyrases, which have both nuclease activity and ligase activity. It will break down the DNA strand. For example, DNA topoisomerase 1, it will specifically act on one strand only. But DNA topoisomerase 2 act on both the strands of DNA. It will break down, then again it will join. That means nuclease activity and ligase activity. That's how they avoid the supercoil formation. If they don't avoid supercoil formation, it leads to distortions of DNA replication be done, is highly detrimental. So avoidation of supercoil or tension generated out of supercoil is most important, and this shall be done by topoisomerases. So in case of, I told already, and what are the differences between prokaryotic and eukaryotic DNA? That means replication. Most of the part, there is a close resemblance between the prokaryotes and eukaryotes, except there are certain differences, like in case of prokaryotes, for better illustration, there is only one origin of replication, whereas in case of eukaryotes, there are many ories. Here DNA polymerase 3 is important enzyme, but here DNA polymerases are different kinds. Say alpha, beta, gamma, delta and epsilon. Delta synthesize the DNA along leading strand and also a proofreading enzyme. Press epsilon, synthesize the DNA along lagging strand, and also a proofreading enzyme. And beta, comparable to DNA polymerase 1 of prokaryote. That means it will remove the RNA primer. RNA primer shall be removed by DNA primer 1 in case of prokaryotes, whereas DNA polymerase beta in case of eukaryotes. Except these two, the process is same. There are Okazaki fragments and others. That means with respect to enzymatic, the nature of enzymes and the number of ORI, that means origin of replications, is different. For example, in eukaryotes, I told you there are many origin of replication. You know what happens? Many replication bubbles. That means multiple replication bubbles, multiple replication forks, because duplication of DNA should be done in a random and rapid manner in case of eukaryotes because of its nature and because of the requirement. The organizational structure of eukaryotes is very complex when compared to prokaryotes. So DNA has to be repeatedly duplicated. During S phase it happens, just for the cell cycle I am saying. This is about today's DNA replication. I told you already it is for the betterment of pool students and particularly for the exams like CSI they may prepare. May share and comment. Okay, we will discuss some other topic in some other class.